All right. So, happy Sunday and very good morning. And today we are going to start with our new topic, that is the central dogma. So, till yesterday, we spoke about certain topics. And in that topics, what we have spoken, a few things are quite important. That is the structure of nitrogenous bases. So, you should know by now why purines are called as purines, why pyrimidines are called as pyrimidines. That is first thing. And the second thing is formation of a polynucleotide chain and formation of nucleotides. Next one is the structure of DNA itself. What are the possible structures that was suggest possible structures of DNA? That is aDNA, bDNA, and zDNA. That is important. And the next one is Chargaff rule, right? Yesterday we discussed about Chargaff's rule. And these are the important things, still what we have discussed for the NEET. Okay. So now we'll begin with central dogma. So this is for understanding, elaborately how the information flow happens in all living organisms. So what is a central dogma? As I said, in all living organisms, DNA becomes a central molecule which holds all the genetic information. So this information is very vital, and having this information does not accomplish everything. So it has to make sure that the information gets passed on down the line. For example, if the cell has to perform a particular activity, such as produce certain proteins, or synthesize certain enzymes, or hormones, or send signals, or protect itself, or you can see give structural integrity, or divide, not divide. So there are like thousands of functions. So to all these functions, somebody has to be informed. So if you're informed, today you attended the class. If you're not informed, you would be like having a good time in the Sunday, right? Similarly, so that pass of information happens through a DNA, what we call, not all of the DNA is having this capacity to do this. Only a certain part of the DNA participates in, what do you tell? Participates in genetics. You can just tell genetics. So this DNA is called as the functional DNA. In simple terms, this functional DNA we call it as genes. So this genes, which is holding the information, is converted into a messenger. Okay, a readable message is created out of this functional DNA, which we call it as RNA. So this RNA comes in several forms, but one of the important message carrying RNA, which we call it as mRNA. So this messenger RNA, which is created in the nucleus, is then transported from the nucleus to the cytoplasm, where the cellular organelles will come and attach to this message, and then they decode this message to construct a protein. Now, the question is, why protein? The protein synthesis happens because, why not lipids? Why not other things? 98% of the biological functions happens because of proteins. You understand? So proteins are real molecules that is responsible for most of the activities that we have today in our living system. So that is why the protein synthesis is everything. So 98% of the biological activities is carried by proteins, and rest of the 2% is by lipids and other molecules. You see? So now they understood that DNA, RNA, and protein is a flow of information. Now we will see how this flow of information, for this flow of information to happen, there are certain processes. For example, how DNA gets duplicated. How does it make copies? How does DNA become RNA? And RNA becomes, RNA is converted into a protein. So all these things we're going to learn from here on, okay? Some technical stuff. So before that, we have to talk about the existence of DNA, how the DNA is present. So if you look at the prokaryotes, so we'll just see how the DNA is present in a cell. So this we call it as a packing of DNA. Our DNA is very, very large. Even for the prokaryotic organism, just we discussed about E. coli yesterday, and E. coli's DNA's length is almost a 4 million base pairs. That's what we talked about. Oh, not 4 million base pairs, 4 million, 4 million base pairs. That's what we talked about yesterday. So if you see this length of DNA to that small cell, you cannot just put it like that, right? So there has to be a certain packing mechanism so that condenses this DNA and puts it in a proper place. So if you look at the prokaryotic organisms, we already know that the prokaryotes do not have any nuclear membrane and they just float around in the nucleus, okay, just like that, okay, in a dissolved form. So here what happens, the DNA is quite not scattered throughout the cell, but they're condensed within a small region. And this region we call it as nucleoid region. Now, here what happens? If you see the DNA, the nature of our DNA, all the DNA that is present, it is quite negatively charged. So now what happens is, to this negatively charged DNA, a positively charged proteins usually comes in groups. So this positively charged proteins in prokaryotes are called as non-histone proteins. Okay, I'll just write it here. Non-histone proteins, or it is also called as scaffold proteins. Scaffold proteins. So the name is quite essential, that is non-histone proteins. So here prokaryotes are quite, what do you tell, primitive organisms. They do not have the complex way to, complex mechanism to pack their DNA because they're the beginning. So that is why these proteins come here, those scaffold proteins or the non-histone proteins come here, and this DNA being the negatively charged, and this non-histone proteins are, or scaffold proteins are positively charged. Now they start winding around this scaffold protein. Now, once they start winding around this scaffold proteins, so they become thicker and thicker threads. So, similarly, which we call it as, these DNA are not really organized as the complex chromosomes, but they are organized as nucleoids. Okay, which the DNA that is organized in the prokaryotes is called as nucleoid. Understood? So that is how the DNA is packed in prokaryotes. So similarly, if you look at the packing of DNA in eukaryotes, it is quite complex. So the prokaryotic, packing happens with a very well organized histone proteins. And histone proteins, what we see here, are positively charged proteins. So here, these positively charged proteins are not just one single protein. So this positively charged protein is a combination, or it is a unit of monomers. So these histone proteins have around 5 monomers, so which we call it as histone monomers. And these histone monomers are labeled here, as you can see in this picture. So they're called as H2A, H2B, and we have H3, H4, okay, and then we have H1 histone. So which is quite important. Now, what are histones? Histones are just proteins, and these proteins are quite positively charged. And the positive charge for these histone proteins comes with the high amount of presence of positively charged proteins, amino acids, positively charged amino acids, or basic amino acids, which we can simply tell, such as lysine and arginine. Very, very important. You can note down this. Histones gain positive charge due to the very rich basic amino acids such as lysine and arginine. In simple terms, what you can think is, if you have a class of 20 and if 15 people are positively charged and if 5 people are negatively charged, the net charge of your class is still positive. Why? Because you have the high amount of positively charged people and very low amount of negatively charged people, so you can rule over them. So that is what happens with the high presence of this lysine and arginine. So this high presence of lysine and arginine will determine or will give this histone monomers the required positive charge. Now what happens? This, with the signals of the cell, now these histone monomers come together and form a ball-like structure, which we call it as histone octamer. Each histone octamer is a ball of 8 histone monomers. You can see 4 and 4, right? 4 and 4. So this ball of 8 histone monomers will form a histone octamer. So once they create a histone octamer, each histone octamer are very well placed, okay, are very well placed at an uniform distance. Now what happens? These histone octamers carry a powerful what charge? Positive charge. Now DNA being negatively charged, okay, it starts winding around this histone octamers, okay, very systematically. Each histone octamer can wind up to 150 base pairs of DNA. Now there is a small tail-like structure. So this tail DNA will just connect from one histone octamer to the another histone octamer, where your winding of the DNA keeps continues to the next octamer. So like this, you can see that the winding keeps happening down the line. Now you can ask, what happens to this DNA? So this DNA, which connects the two histone octamer, is called as linker DNA. And it is held by a special type of histone protein that is called as H1 histone protein. So the H1 histone protein holds this DNA called as linker DNA that connects the one histone octamer to the another histone octamer. Now we understood the packing of the DNA has accomplished by formation of histone octamer and winding of the DNA around this histone octamer, and each histone octamer can wind up to 150 base pairs of DNA. Right? Now what happens? Now, once we have this histone octamer, now this structure, when we look at an electron microscope, we can see that a beautiful pearl-like structure, pearl chain-like structure, right? So here they're quite organized like the beads on the string, or we call it as beads on the string model, or it is also called as nucleosome. So this structure, when we observe it under a microscope, which appear like this, is called as nucleosome. So once we have this nucleosome structure, now what happens? So these are just like a quite thicker threads. Now, chromatin proteins or non-histone proteins, they come and attach to these regions of your histones, and then they condense them even more further. So the condensation happens in such a way that these bead-like structures, they're further converted into thicker thread-like structures, and these thread-like structures, which is a repeating unit, and these are called as a chromatin fiber. So these chromatin fibers are further condensed with the help of chromatin proteins to form a chromosome. Now, this coiling of this chromatin to form a chromosome happens at a certain stage of the cell division, and this cell division stage is called as metaphase. So this happens at metaphase. Now, when you look at the X-ray of your chromosomes, what you can understand is, if I take an X-ray of my chromosome and observe it under a microscope, what I can see is my chromosome, like which appears, somewhat like this, right? Somewhat like this, do not have a uniform color. At some places, like a zebra, they have very dark bands, and in some places they have relatively light bands. Like similarly, if you take an X-ray of your hand, you can see that the bones appear quite dark, and then you can see that the muscle and tissues are quite light. Similarly, you have the dark bands and the light bands that appear in the chromosome. Now, what are these? Here we can understand that the dark band and the light band is because of the amount of packing that has happened within the chromosome. The dark band is because there is a very tight packing that has happened at this region, and this very tightly packed region, we call it as heterochromatin region. And many of the scientists argue that this heterochromatin region is due to, the presence of non-functional DNA. And they are the large amount of DNA which you can see in the chromosome. They present in a very large quantity. That means heterochromatin region is very large compared to that of euchromatin. Now, why do we have such a large chunk of DNA which is waste? They say that this is either ancestral DNA, which we have lost, the characteristics which we have lost during the course of evolution, or this DNA is also expected to give structural integrity or structural support to our chromosomes while movement of chromosomes or while binding of chromosomes, etc. Now, the rest of the region, which is a lightly light packed region, which we call it as euchromatin region, is one that has the genes or the functional DNA. So this is a very active DNA that is then participating in the formation of messenger RNA, and then it gives rise to a protein. Understood? Euchromatin participates in genetic flow of information. So we understood what is heterochromatin, what is euchromatin, and also we understood the rest of the facts about packing of DNA. Now let us go for the next. Our next topic is the search for genetic material. Now this is a very, very fascinating topic. Why? Because the text, it is so well organized for you people to understand to understand the procedure, scientific procedure for doing an experiment. It's very, very important. It's very, very important for you to learn this the process or the procedure of experiments. It's like mini scientists. So let us see what we are trying to learn here. Why search for genetic material has to happen? In spite of learning the structure of DNA, in spite of learning all this till now, we know the really what is DNA made up of, what is DNA's capabilities, and how it is packed. We know everything right now, but now why are we studying the search for genetic material? Because back then it was not known. Now, whatever the known facts that we had already is just given here in a textbook. So priorly, our ancestors did not know, or our scientists did not know before. So how did they come to know that the DNA is a genetic material? So that is what we are trying to learn. So here in the search for genetic material, it starts from the transforming principle, the famous transformation principle, and a brilliant experiment which is conducted by Frederick Griffith. So Frederick Griffith conducts this experiment to show that there is a genetic material that is transferred between the two bacteria. Now, before we learn this experiment, there are few facts that you have to understand. The first fact is what happens during the unfavorable conditions in bacteria. During unfavorable condition in bacteria, they try to show a type of pseudosexual reproduction. We know that they undergo strictly asexual reproduction, but we call it a pseudosexual. Why pseudosexual? They mimic a type of sexual reproduction, but it is not truly a sexual reproduction. So this type of pseudosexual reproduction, which we call it as conjugation. Okay, we call it as conjugation. So this conjugation is a process. So what happens in conjugation? Maybe you know because you learned in the biological classification. But if you know, it's good. If you don't know, listen. Now we have, let us say that we have two different types of bacteria. And among these two different types of bacteria, I will name one type of bacteria as positive and I will name another type of bacteria as negative. So now what happens is they have a type of genetic material outside their outside their nucleus. So whenever a DNA is present outside the nucleus, we call it as extra chromosomal DNA. And here bacteria have an extra chromosomal DNA, and this DNA is called as plasmid. Okay. So this plasmid is very, very important for bacteria because it gives rise to so many different characteristics that the bacteria has. Characteristics are very, very important, right? Now what happens? An unfavorable condition comes, such as heat, such as dryness, or whatever it is, or antibiotics is put on this bacteria to kill them. Now what happens? Let us say that this negative bacteria has a certain character. So this negative bacteria will start making two copies, one extra copy of this DNA. And here, these two cells come close together, they form a bridge, and this extra copy from the negative bacteria will start flowing into this tube called as conjugational bridge, and finally it reaches the positive bacteria. This, after reaching the positive bacteria, this DNA will become a part of the bigger DNA or the bigger plasmid DNA, and finally converting this positive bacteria into positive bacteria into a negative bacteria, right? So this process is called as conjugation. Now, why did we see? Why did we learn about conjugation in this experiment? I'll talk about it. Okay, you'll have to understand this. Now, what happens in this experiment? Griffith actually chooses two important organisms for this experiments. One is a mice, okay, a rat, and the other one is a bacteria. So this bacteria he chooses is called as Streptococcus pneumoniae. So the Streptococcus pneumoniae bacteria is the one which causes a disease called as pneumonia, which causes a pleural infection in your or the infection in the alveoli of the lungs. When they cause an infection in the alveoli of the lungs, so they basically what happens? So they create this pneumonia and you have the breathing trouble and finally you die. So what happens? Not all the bacteria are bad. Even in Streptococcus pneumoniae, they have two divisions, two categories. One which has the capacity to cause disease and one which do not have the capacity to cause disease. So this capacity to cause disease is called as virulency or virulent strain. So now what happens? There is a glycoprotein capsule that is present around this disease causing bacteria, which makes it look very, very smooth. So we call this bacteria as smooth strain bacteria. So the smooth strain bacteria is the one which has the capacity to cause a disease. So that is why we call them as virulent bacteria. Understood? Or virulent Streptococcus pneumoniae. And the other one is a non-virulent bacteria. Because of the lack of this capsule, their colonies or their structures look quite rough when we observe it under the microscope. So the other one is called as non-virulent strain or non-virulent variety, or also called as rough strain. So the rough strain do not cause a disease, whereas the smooth strain causes a disease. And as I told you before, the other organism is the mice. So here is what happens. Now, Griffith takes a smooth strain, okay, live smooth strain, that is living bacteria, and he just injects into the mice. The mice develops pneumonia and it dies. So obvious. Next, he takes a rough strain and then he injects into the mice. The mice do not get pneumonia because the rough strain do not have the capacity to cause disease. So the mice live happily. Now what he does? He takes the smooth strain and he does it heat killing. Now, many students just get confused. What is heat killing? And heat killing, somebody like us, non-scientific people, which we don't know much about science, we are just learning, right? So what we think, we're just like boiling. Let's boil the bacteria. No, we are not boiling the bacteria. We're just heating. So what's the difference? We just heat the bacteria to around 40 degrees Celsius. So now, just increasing this rapid temperature, there are some significant changes that are happening within the bacterial cell. So the sudden raise in temperature will affect this bacteria in such a way that it will lose the certain characteristics. I will tell you how. So just heat kills it. When he raises the temperature suddenly for certain times, and this bacteria will be living, but it will not have certain characteristics. Now what happens? The heat kills this bacteria, which bacteria? Smooth strain, and then he injects into the mice. When he injects into the mice lives. What happened? When he heat killed the bacteria, certain proteins which were helping this bacteria to cause a disease got destroyed. Because when you increase the temperature, the protein gets degenerated. Right? So when the destruction of this protein happened, this bacteria, lost this capability to cause the disease. And finally, when it was injected in the mice lives. So this process of, making a infectious bacteria into non-infectious bacteria is called as attenuation. So this attenuation process we just saw. What is one process of attenuating? Just heat kill it. Increase the temperature, it will lose the proteins and it will not be having the capacity to cause the disease. Simple. Now, what happens? Once this heat kill bacteria or heat kill smooth strain was injected into the mice was living happily without developing the pneumonia. Now, what Griffith does is he takes the same heat killed smooth strain and then he mixes with the rough strain. No, he puts it. He takes some of it and he puts it in the rough strain bacteria. Once he puts this heat kill smooth strain into the rough strain bacteria, he leaves it for some time and then he just takes some bacteria and he just injects into the mice. Now, what do we call this? Heat kills smooth strain plus rough strain and then he injects into the bacteria. Sadly, the mice dies. Now, what happened here? Now, Griffith didn't know what happened, but we know what happened. Now, the smooth strain bacteria had the DNA very intact, or the genes that was responsible for causing the virulency, it had it. The rough strain did not have it. Now, what happened? The smooth strain makes copies of it and sends it to the rough strain. The rough strain did not have the DNA, but the rough strain had all the proteins for it. So once this DNA got incorporated into the rough strain, the rough strain became smooth strain. So what happened? Now this caused the disease to the mice and sadly mice died. Now this transformed the rough strain into the smooth strain. Now, sadly, the Griffith could not tell what was transferred between the smooth strain to the rough strain. They could only tell that there is something that is passed on, and information got passed on from the smooth strain bacteria to the rough strain. That is what killed the mice. So when he did not conclude what happened, that experiment, one of the famous experiment, for the first time people saw that the transfer of character took place between the two different organisms, two different varieties of organisms. So this was the advantage. He showed that there is something transformed, but he could not conclude what is transformed. The disadvantage is that one, what he could not conclude what was transformed. So this is called as a famous transformation experiment of Griffith. Next, we'll go for the next one, right? What is next one? Somebody came up and they redid the work on Griffith's experiment, which you call it as biochemical nature of the transformation experiment. Very, very important experiment, students. So let's see what is happening here. So in the biochemical characterization of transformation experiment, what they're trying to do is they're trying to see what really got transformed in the Griffith's principle. And the people who worked on this biochemical nature of what got transformed between them are Oswald Avery, very, very important, these scientists, okay? Oswald Avery, Colin McLean McCarthy. Or Avery McLean McCarthy experiment, it is called as. So these three scientists, they worked on what they wanted to see, what are the biochemicals that got transferred in this process. So what they did was very, very brilliant experiment again. They understood that there are enzymes that can digest our biomolecules. So the possible candidates they thought to become a genetic material was three. What are those? Either protein should have been transferred between the two, or it should be DNA, or it should be RNA. So to analyze this, what they did was they wanted to purify this biomolecule. So what they did was to purify the biomolecule. How do we purify a biomolecule? We use digestive enzymes. So what are digestive enzymes? Something that breaks down certain biomolecule. So now what happens? In this experiment, they takes a test tube, and each test tube has heat-killed smooth strain. Heat-killed smooth strain was there. Now, these people, they choose the digestive enzymes in such a way that we have three candidates. What are those? Proteins, and then we have RNA, and then we have DNA. For that, they choose three different enzymes. What are those? One is proteases, next one is RNases, and DNases. Proteases will digest proteins, RNases will digest RNA, DNases will digest DNA. Understood? Now what happens? They take three test tubes. One test tube, they have heat-killed smooth strain. To that, they add proteases. When they add proteases, all the proteins get destroyed. Next one, to another test tube, they take the RNases and then they add RNases to the heat-killed smooth strain. Then all the RNA gets destroyed. Next one, we have the DNases, and for the final test tube, they have the heat-killed smooth strain. For that, they add the DNases. Here there is no protein, here there is no RNA, and here there is no DNA. Three different test tubes. Test tube 1, what is that? Heat-killed smooth strain plus proteases, and then they mix it up with the live R strain. When they mix it up with the live R strain, 1 second, okay? Sorry. Yeah. When they mix it up with the live R strain, now what happens? Transformation of R strain to S strain happens. Why? What is transformed here? DNA is transferred, so destruction of protein will not matter. Next one. They take the heat-kill smooth strain and they add RNases and then they mix it up with the live R strain. When they do this, again the transformation takes place because RNA got destroyed, no problem. Now the DNA is transferred, so that will help the bacteria to get transformation. Next, heat-killed S strain plus DNases plus R strain when they added, now the transformation will not take place. Why the transformation will not take place? The DNases destroyed the DNA of heat-killed smooth strain. When it destroyed the DNA of heat-killed smooth strain, I told you in the prior experiment that the DNA was the one that moved between the heat-killed smooth strain to the rough strain. That we knew, but they don't know, right? Back then. So when this happened, when the DNases destroyed the heat-killed smooth strain's DNA, now it didn't have any DNA to transfer or send it to the R strain. So the R strain did not get transformed and the mice will live. Understood? So this showed that, oh, this was a solid experiment that proved that the DNA is a genetic material. Unfortunately, what happened, students? Unfortunately, this particular experiment was not widely accepted. Many of the biologists, they thought that this experiment was not convincible. Why? Simple reason, very, very simple reason. You laugh at this reason. They thought DNA cannot be an ideal genetic material because it is too simple. Why it is too simple? I told you, it is made up of only 4 different types of nucleotides. How can just 4 different ATCG can determine so much diversity and so much of this biology? Just 4 molecules? It's not possible. So what they thought was proteins are the one. Okay, other biologists thought that proteins are the one which are the ideal genetic material. Their argument was very simple. They thought that proteins have so many different types of amino acids, 20 amino acids are there. So DNA has only 4 different types of nucleotides. So the combination of these amino acids are large number compared to the combination of ATCG. So that is why they thought that protein might be the genetic material and they were not convinced. So that is why this experiment did not get the required attention that it had to get. Okay, so this was simple. It was such a simple experiment to prove that DNA is a genetic material. Simple, gone. But it was not accepted, unfortunately. Now, again, one more experiment is designed by, after so many years. You see the number of years that they had to wait. 1933 to 44. Here you see. It took almost 1952. Almost 10 years. Okay, 10 years of wait happened. And then again, two more brilliant scientists comes. So here they proved that DNA is a genetic material. Who are those famous scientists? This experiment is famously known as Hershey and Chase experiment. So their full name is Alfred Hershey and Martha Chase. We call it as Hershey and Chase experiment. Okay, so that it is very easy for us to remember. What happens in the Hershey and Chase experiment? Very, very beautiful experiment. So they work with viruses. So in Hershey and Chase designed this experiment. In this experiment, they use 2 organisms. One is a virus, that is a bacteriophage, and other one is a bacteria, E. coli bacteria. Now, what happens in this experiment? In this experiment, what they do is they take this ideal organisms, that is bacteriophage and bacteria, and they play with it. Okay. So what happens? Before we learn this experiment, we have to understand something about this virus bacteriophage. They infect bacteria. How do they infect bacteria? So the bacteriophage is a very cool and weird looking virus. At the same time, I like this virus so much because of its appearance. It looks like a robot from the end of the world, Wars of the World movie. Very cool, right? So this bacteria has a head, neck, and a tail region. So this head region has a special package. In this package, we call it as capsid. Okay, so this capsid is very, very crazy. Why? Because it has a genetic material, DNA. So they have DNA in it. So within this capsid are special proteins also, which help this capsid survive within the bacterial cell. Now what happens? This virus comes and attaches to the bacterial cell, drills a hole into its cell wall and the cell membrane, and then injects this capsid into the bacterial cell. Right? I'm telling it as simple as possible so that you understand. Now, once it injects this capsid, the capsid opens, the DNA gets released. This DNA becomes the part of the DNA of bacteria. Now, it controls the whole motherboard of the bacteria, right? It completely controls its software. DNA is a software. So once it becomes the integrated part of the bacterial DNA, now it tells, make copies of me. That is what the proteins come. The proteins come and attach, and they keep on helping this virus to create more and more copies, extra, like photocopy machine. It will create more and more copies of this DNA. Each DNA copy can become a new virus. So each bacterial cell become a bacterial cell will become a factory of bacteriophage. So that's how it controls the bacterial cell, and that's how it survives within the bacteria, finally killing and destroying the bacteria. So this is a very nice experiment, right? So we know how the virus survives. Now let us get back to this experiment. What happens in this experiment? So first, they prepare 2 sets of experiment. Very brilliant people. Why? So they thought that the possible candidates for becoming a genetic material, as our biologists believe for a very long time, what are those? One is a DNA, right? Other one is a protein. So they understood, very simple, the biochemical nature of these two. They thought that DNA has a prominent phosphate end, right? They have a phosphate molecule. The proteins do not have. Whereas the proteins have a prominent sulfur molecule. So this sulfur appears in proteins in the bridges, right? They attach these polypeptide chains together. So sulfur is present in the proteins and phosphate is present in the DNA. So that is what transformed this experiment. So what they did was, if you guys think that the proteins are the genetic material, that means a sulfur should be present. I will tell you about it. If you think that the DNA is a genetic material, then we are trying to target the phosphate. So what happens? They labeling will happen. So what do they label? They label in the first experiment, they take bacteriophage, and to this bacteriophage, they label it with a radioactive sulfur. What is this radioactive sulfur? It's an isotope of sulfur. Usually the sulfur that we see naturally occurring is 32, S32. But here the sulfur they have used is S35. So the isotope of S35 is radioactive. So here, when they use the radioactive sulfur and they label the bacteriophage, now you know that the virus structure is in such a way that all this head, neck, and tail region, whatever we see, right, is made up of proteins. So their entire body gets radioactive apart from the DNA. The DNA will not get affected here. Now what they do is they take this radioactively labeled sulfur bacteriophage and then they infect the E. coli cells. They leave it on the E. coli cells. They'll mix it with E. coli cells. They're happy. They got a host. So they attack this bacterial cell and then they inject their genetic material. Now, once they inject this genetic material, now what happens? They are still attached to the surface of the bacterial cell. Now, what we do is we take these cells and then we blending. We do the blending. What is blending? Just like you make the milkshake of or the juice, fruit juice. You just mix it up. When you mix it, now this bacteriophage which was attached to the cell surface will just fly out. Now we take this solution and then we put it in the centrifuge. When we put it in the centrifuge and spin it, now what happens? It will just create two layers. What are these two layers? One layer which is a solid, which you call it as pellet. Okay, so this layer is called as pellet or pellet. So this pellet contains bacterial cells. It contains bacterial cell and bacterial DNA, right? Now, this supernatant or the liquid part contains proteins. What protein? Viral proteins. Okay, it contains what? Viral protein. Now, when they labeled it with radioactive sulfur and then they blend it and then they saw the test tube, they saw two regions. One is a pellet region, other one is a supernatant region. Here, what happened? The radioactivity was detected in the supernatant. When the radioactivity was detected in the supernatant, what is there in the supernatant? Only viral proteins are there. Only viral proteins are there means there was nothing, no radioactivity that was transferred between the virus and the bacteria. So that proved that the protein is not the genetic material. In case if it was transferred into if it was injected into the bacterial cell, obviously the bacteria, the pellet should have been the radioactive. Here the supernatant is radioactive. So that proves that protein is not participating. Now, how do we know this? We'll see the next experiment. Similarly, how we saw in the last experiment, experiment 2 was they take the bacteriophage, they label the DNA or they label this cell, they label this bacteriophage with radioactive phosphorus. What is radioactive phosphorus? The normal phosphorus is P30. Okay, radioactive phosphorus is P32. Okay, I already got the P, right? I'll just erase this. It is P32. So you remember this isotopes. So when this P32 was labeled with this bacteriophage, you know that phosphate is in DNA. So the DNA will become radioactive. Now they leave it to infect the bacterial cell. The bacteriophage infects injects its DNA into the bacterial cell. Now it will become a part of bacteria. Now you take this and then you centrifuge, blend and centrifuge. When you blend and centrifuge, you get two regions again. What are those? One is a supernatant region, other one is a pellet region. This time, supernatant did not have any radioactivity, whereas the pellet was showing the radioactivity. Now I told you what does pellet contain? The Pellet contains bacterial cells as well as the nucleus of bacteria and the DNA of bacteria. Now the pellet started showing the radioactivity. That means the radioactivity of the bacteriophage was transferred into the bacterial cell. Now the bacterial cell is showing the radioactivity. Now what was transferred? Not the entire virus. The virus was injecting only the DNA. So the DNA became radioactive here. So that proved that DNA is a genetic material. And this experiment that proved the genetic material is the DNA is called as the famous Hershey and Chase experiment, right? Okay, so are you guys understanding? Yes. I put a technical check. Oh, sorry, a poll. You have a poll on your screen. A, I understood. B, I did not understand. Okay, give me the answer soon. Come on, guys. There should be equal rights, right? For the past 45 minutes, we are talking. So I want you guys to give me a vote, conveying me that yes, I am understanding, or no, I did not understand. Is this A and B? You slept, one of you? Faster. Okay. A, you understood. B, you did not understand. Or is my voice audible? Now you're confusing me. Students, respond. Soon. Make it soon. Come on, this is a simple question. Poll. I'm not even asking you to type. Just poll. Press either A or B. Understood A, did not understand B. Simple, right? Students on the YouTube, you guys can put it in the chat box if you are like trying to understand this. If not, let me know. We can just redo it. Okay. Chalo. Okay, let's go ahead and I will end it. Okay. Assuming that you guys are understanding. So, not my problem. You talk, it's better. You don't talk, it's very bad. Chalo. Now, since we spoke till now, and the next topic is, finally it was proved that DNA is a genetic material, and it comes to one more important criteria. What is that? DNA is a very, very beautiful molecule, right? So, what makes it one of the most wonderful molecules on our planet? Now, the DNA's capacity is fascinating. Why it is fascinating? Just look around you. Just look at yourself and your friends which are beside you. So if you see the amount of changes and the amount of differences and the exuberant nature of the DNA, it's quite, it will make you shut your mouth and just observe and just accept the beauty of this. Now, what makes it such a beautiful molecule? First thing, whenever we call it as a genetic material, it has to have certain characteristics. Like if I tell this is a good boy, is a bad boy. Now I will be trying to judge him on a certain criteria, right? Similarly, for biologists, if I have to tell that, okay, why not lipid is a genetic material? Why not minerals? Certain minerals are genetic material. Why not RNA becomes an ideal genetic material? Something like that. So I have a criteria. We have a criteria. What is the criteria? First thing, it should be able to generate its copies, or it should be able to photocopy itself. Why this is important? Now look at you are made up of 30 trillion cells. Now the number of cells is so huge. All of the cells should have the same genetic material. We know that it is a mitotic division. Imagine that each of your cell has different genetic material. Different genetic material, different software. If different software, different function. Now, if this happens, would your body just behave as one? Not possible, right? Similarly, when we talk about the meiosis division during the gamete formation, now you see that so much amazing organisms are created. Now, if the transformation of characteristics do not take place from parents or to offsprings, when they create these gametes, would it be possible for speciation to occur? Would we create a species like elephants, like elephant species, or if you have tiger species, are the species to continue? Would it be possible? Definitely no, it's not possible. That is why the replication is a very important criteria, and it should be able to make copies itself. Next one, it should be chemically and structurally stable. Very, very important character. Now, what is chemical stability? It should not be reactive. It should not change its form now and then, like our RNA. RNA is very, very, very dynamic, always changing. That's why RNA is a catalytic molecule more than the stable genetic material. Example, if you just see our HIV virus or your COVID virus, why it was very hard for us to create vaccine? Instability, because it is RNA virus. It has genetic material as RNA. So much instability. It is always changing from one generation to another generation. And it is not at all identical to their parents. So that is what makes them very vicious organisms. Now just imagine that you're a human and suddenly you're somebody else, your son or daughter is something else, having different characters. So that is called as instability, chemical instability. Next one is structural instability. What is structural instability? Now, if you look at our DNA has deoxyribose sugar. And our deoxyribose sugar is very, very nice. Why it is very, very nice? I say because it has hydrogen in the second carbon atom and OH in the third carbon atom. You see, this is deoxy. Okay, I'll just write it as DNA. If you look at RNA, RNA also has a pentose sugar, and this pentose sugar we call it as ribose sugar. Here in the second position also there is OH. In the third position also there is OH. Now, what did I tell you about formation of polypeptide chain? Which is fifth phosphate group, and the third OH group participates in DNA. Whereas come to RNA, now the phosphate group can either participate with third carbon atom, or it can participate with second carbon atom. You understand? So there is an additional OH in the second position, which makes it structurally unstable. Like the zigzag, it can go like 3, or it can go to 2, or it can go to 3, or it can go to 2. So this will make it not uniform. So we want a uniform and structurally stable molecule, and the DNA presents a beautiful structural stability. Next one, it should provide slow changes in the form of mutations and recombination. Now, the sudden changes, this is not slow mutation. Fast changes is mutation. Slow changes is recombination. We're wrong, fast. Now, what is recombination? Appearance of non-parental characteristics. We studied in principles of inheritance and variation, right? Very, very slow. Now, you just compare yourself with 10 generations before. Now, what you can see that, oh, we are not the same. They have certain characteristics that we don't have right now. Why? As the earth changes, as our environment changes, our food changes, our resources changes, the living style changes. And if we just stick on to the same genetic material, we cannot adapt, we cannot change, no adaptation, no survival. Darwin said, survival of the fittest. And who is the fittest? The one who adapts is the fittest, correct? So these slow changes in the form of recombination and sudden changes in the form of mutations are two essential factors that promote evolution. And the promotion of evolution is what continues survival of species. Understood? So that should happen. Next one, it should be able to express itself. Expression is so beautiful. Now, this expression happens through formation of RNA, formation of proteins, and these proteins build characteristics, and these characteristics are expressed in the form of phenotypic and genotypic characteristics, right? So those are called as Mendelian characteristics. So Mendelian characteristics we already know, we can see them, we can understand them. So this expression has to happen. Next one, we know that the protein is not at all fit and it does not fulfill any of these criteria, so it is not the genetic material. Whereas when it comes to the two candidates, there is DNA and RNA. Both of them fill the criteria, and they both are genetic material. And understand this point: all living organisms majoritarily have DNA as a genetic material, whereas coming to RNA is seen as a genetic material in viruses. Even they at one point convert themselves into DNA for expression. Understood? So this is about if the DNA is a genetic material. Next one. Coming to the replication of DNA. What is replication of DNA? Replication of DNA is nothing but making photocopies of DNA. You have a parent DNA. Okay, parent DNA means, for example, you have a parent cell. It divides. It makes two daughter cells. If it makes two daughter cells, even its genetic material, that is DNA, has to get into the two daughter cells, right? How did it become two? One became two. So this is what we are trying to study in replication of DNA. Now, how does it happen? Now, Watson and Crick, back in the days, they suggested that the possible way in which the DNA is making copies is through semi-conservative mode of replication. But they did not have any tools, or they did not build any scientific experiment to prove that the DNA replicates semi-conservative. So that is why their idea was not quite accepted. Semi-conservative mode of replication. We'll understand what is semi-conservative mode of replication. So usually the DNA replication happens in the nucleus, and this particular phase, called as the S phase of the cell cycle, that is where the DNA replication happens. And usually the DNA is present in the nucleoplasm, which we know already. Now, if you see the DNA replication, back in the days, they thought that there are three ways in which the DNA can replicate. And very, very important concept, you have to concentrate. Last one, okay? So here, if you see that, they suggested that there are three modes of DNA replication. One is conservative, next one is semi-conservative, other one is disruptive. Now, what is conservative? Conserve means save, or just save, okay, in simple terms. Conservative, what is happening means, you see there is a parental DNA. The parental DNA, this is the parent cell, it is a daughter cell. This is the parental DNA. So this parental DNA goes to this cell. What happens to the other cell? This cell gets a brand new DNA. You see this red color, this is a brand new DNA. Using this as a reference, it will construct a brand new DNA and then it puts into the other cell. So 100% of the parent gene is conserved or passed on to the next cell. That is what is called as conservative. You understand? So in another mode called as semi-conservative, semi means what? Half. So half conservation. What happens here? This is very, very widely accepted mode of replication. Here, what is happening? You take one parental DNA, use this as a template. Template means reference. Okay, use this as a reference, construct another strand of DNA. We know that our DNA is made up of two strands, right? So one will be parent, other one will be a daughter. Again, you take this DNA as a template and construct a new DNA. So this is called a semi-conservative mode of replication. Understood? Next one is disruptive. What is disruptive? You take one DNA, you take parts of the parental DNA, and you construct a small new DNA. You understand? You take a fragment of parental DNA and you fill it up with new DNA. So this is called a disruptive mode of replication. And as the time passes, you can see that the disappearance of parent DNA takes place. Here, as the time passes, here also the disappearance of parent DNA takes place. Correct? So this is what is called as disruptive mode of replication. So three modes of replication: conservative, semi-conservative, disruptive. And you should know the diagram, which is what? Okay. So we will understand how they proved that the DNA replicates semi-conservatively in the next class. Okay. So next class, we will try to finish more and more topic, because you need this little bit of brush up before we solve the questions. Okay. So we have finished a significant amount of portion. Next comes replication, transcription, translation. We will finish it off. And then all we will be left with is questions, and we'll directly go with questions because the rest of them is factual, which you can easily understand. Okay, we'll pick up selective topics for the next case. All right, students. Thank you very much for attending today's session. I'll see you in the next weekend. If you have any doubts, you can put it in the message. If not, you can leave the class. All right. Have a nice day. Yeah, sorry. Have a nice day. All right, students, I'm gonna end the session. See you. Bye bye.